A very good afternoon to everyone. So myself, Dr. Dilpreet Singh, working as an associate professor and your subject teacher in the B Farm sixth semester, that is biopharmaceutics and pharmacokinetics. In the previous lecture, we have discussed about the absorption, what is the mechanism of absorption, what is the absorption pathways, and what is the factor affecting the drug absorption. So in the second part, we have discussed about the distribution, what is the effect of the distribution on the pharmacokinetics of a drug, what is the effect of distribution on the VD clearance and the apparent volume. So now we have shifted towards the advanced version, that is called determination and kinetics of the protein binding. So basically, when we talk about the binding of the drugs, so the amount of the drug binding to plasma protein is governed by the measuring the free active fraction that has the important factor on the pharmacokinetics and pharmacodynamics of a drug. So in this lecture, we have discussed about the methods to determine the protein binding and what is the kinetics of the protein binding, including the direct methods and the indirect method and various theoretical simulations used to calculate the protein fraction and non-protein fraction. So when we talk about the determination of the protein binding, so basically the binding is always be reversible. When we talk about the binding on the drug and the protein, it always be reversible, and it is commonly stated that there is a specific transport system when we talk about the free drug molecules that are able to cross the biological membrane. So if we talk about the membranes, they can be distributed to tissues, undergo metabolism and the glomerular filtration. So there several types of the drugs, for example, like the fluoxetine, flurbiprofen, and ketoprofen, they are bind with the catalytic site of the enzyme to form a reversible protein complex, which is highly be unstable, so it gets rapidly excreted in the urine. For the larger binding molecules, we have the protein molecule or peptide molecule. The binding is always be reversible, and the bound fraction is slowly being detached from the protein molecule, and it get released slowly in the systemic circulation for therapeutic action. So such type of the therapeutic effect is longer in our body. So there is chances of toxicity and accumulation in the presence of some protein molecules. So there is a need to determine the protein drug binding in a biological system. So there are two types of the methods available for protein drug binding. That is called indirect technique. So indirect technique, it involves the separation of the bound protein molecule from the free micromolecules using the ultrafiltration or ultracentrifugation. So using that high shearing force through centrifugation or filtration, we can separate the bound fraction from the unbound fraction, and we can determine by analytical techniques like the UV method or the like spectrophotometry is there, and AAS is there, atomic absorption spectrophotometer method. So there are a lot of the methods that are available in the literature to determine the bound concentration. So but firstly, we have to separate the bound form from the unbound form. So in the direct techniques, so we do not need to separate the bound form from the free macromolecules, and it can be determined by measuring the active concentration, that is bound concentration of a drug from a protein molecule using analytical techniques like UV spectrophotometer. So if we talk about the factor affecting the protein drug binding, so basically it is divided into three major categories, that is called physicochemical properties, physiological properties, and the drug related properties. In drug related factors, the first is the physicochemical characteristics of a drug. So protein binding, when we talk about the lipophilicity, it is directly related to what we call as the improved solubility or improved hydrophilicity. So an increase in the hydrophilicity, or we call as a decrease in the lipophilicity, it decreases the extent of binding. So if we talk about the increase in the lipophilicity, so the more lipophilic drug, it causes the increase in the extent of binding, and the bound fraction is unavailable for absorption. But that is the basic reason behind that. The bound fraction is unavailable absorption because of the high lipophilicity. So lipophilicity, it creates a hindrance to the lesser chances of the drug available for the systemic circulation. For example, fluoxetine. Fluoxetine is having high lipophilicity, it having the 95% protein bound. So basically, it is unavailable for the absorption. So basically, it causes the erratic bioavailability. Ampicillin, it is basically hydrophilic in nature, so it having the 20% of the protein binding, so it having unbound fraction largely available for the absorption, so it getting the improved or uniform bioavailability. A other example we can study like the thiopental. Thiopental is basically highly lipophilic in nature. It binds with the adipose tissue, protein molecules, or some substrate molecules to bind with the active side of the enzymes. When we talk about the penicillin, so penicillin basically a acidic drug. It binds to the human serum albumin. Basically, HSA is basically a basic glycoprotein or alpha acid glycoprotein. Basically, it has the molecular weight of 40,000 daltons. So majority of the acidic drug it binds to the HSA, including penicillins. Imipramine, that is basically basic drug that it binds to alpha acid glycoprotein, that is another class of proteins. Unionized, it binds to the lipoproteins. So this effect you can largely be measured that the hydrophilicity or lipophilicity or the physicochemical characteristic it can play a major role in the binding of different proteins. It also affects the extent of binding. It also affects the type of the protein which need to be bound to a particular drug. So that can impart a major effect on the circulation. B is the concentration of the drug in the body. So the alteration in the concentration of the drug substance as well as the protein molecule or surfaces, it subsequently bring alteration in the protein binding process. So if the alteration in the protein binding stress is a process is the given, so there is a high chances that the concentration of the drug substance as well as the protein molecule is subsidiary or is subsidized in the body for longer period of the time. So the longer period of time, it causes the accumulation or toxicity in the body, which doesn't get been replaced or replenished during time. So we can detect the last plasma concentration if the concentration of the drug in the body is very high, and also the protein binding is very less. So depending upon the factors, depending upon the type of the protein, depending upon the type of the drug, depending upon the concentration of the drug in the body, we can depict the alteration in the protein binding process or alteration in the concentration of the drug in the free form or in the active form. C is the affinity of a drug for a particular binding component. So this factor is entirely depends upon the degree of attraction or degree of ionization or the affinity of the protein molecule or the tissue that moves towards the moiety. Basically, when we talk about the unionization or ionization, so basically ionized form of the drug having greater affinity for a particular binding component. For example, the lidocaine. So lidocaine basically is a weakly acidic drug. It can convert it into its basic form in the basic pH, and that it having that ability to properly ionize the molecule, and the active ionized drug it having greater affinity for binding with the alpha acid glycoprotein than that of human serum albumin. Basically, it converts. So basically, the degree of attraction. I have said that lidocaine is basically a weakly acidic drug. So it will convert into its ionized form in the basic pH. So it having more affinity for alpha acid glycoprotein. So most of the basic molecules alpha that can bind with the AAG than SSA. So alpha acid glycoprotein that having greater affinity for completely ionized molecule than that of ionized molecule because AAG is produced a unstable or weak complex. So that weak complex is being dissociated over time and it gets been eliminated throughout the body. Second type is the protein or tissue related factor. So when we talk about the tissue related factor, so first description is the physicochemical characteristics of a protein or a binding agent. So lipoproteins and the adipose tissue that having the tendency to bind the lipophilic drugs by dissolving them in a lipid core. So basically, when we talk about lipophilic drug, its solubility it more towards in the lipophilic environment or in the lipidic environment or the phospholipid environment. So they have the ability to dissolve them in the lipid core. So it having that concentration more in the protein fraction. So it been distributed highly in the tissue compartment. So it will more rapidly distribute in the tissue compartment. So there are less chances of excreted unchanged form in the urine. So its reabsorption is initiated when we talk about highly lipophilicity. B is the concentration of the protein or the binding component. So among the various plasma proteins, it binding being predominantly occurs in the albumin as it is present in high concentration in comparison to other plasma proteins. The amount of several proteins and the tissue component at available for the binding, it changes during the disease state. So when we talk about the inflammation conditions, when we talk about the nephritis, or when we talk about the liver metabolism, so there are the chances that protein concentration can vary with patient to patient. So the binding affinity that can vary more in the albumin or more in the alpha acid glycoprotein, because these two proteins they can alter in the disease state at very high ratio. So when we talk about the concentration of the protein, so the amount of the several protein and the tissue component available for binding, it changes during the disease state. So number of the binding sites. Albumin has a number of binding sites. As I've already discussed, it has four binding sites. That is called warfarin binding site, diazepam binding site, digitoxin binding site, or tamoxifen binding site. So if that binding site is really really high, so it can causes the displacement interaction. So it causes the increased free drug concentration. That causes the adverse effects. So several drug it can bind to more than one binding site, that is called competitive inhibition. So one can displace the binding of the other drug. So the displaced drug concentration is high in the body, that causes the accumulation or toxicity. Third is the drug interaction. So the competition between the drugs for the different binding site is called a displacement interaction. So basically, if the drug is bind to a particular protein or it having affinity for a particular protein, so the drug to it having that similar covalent pocket or similar substrate pocket that having the similar binding affinity to that of particular protein, it displays the drug one and it can bind with that protein and then for the therapeutic action. So D1 is a displaced drug, D2 is the displacer drug. So the administration of phenylbutazone to a particular patient on warfarin therapy result in the hemorrhagic reaction, because the warfarin has a competitive binding site. So phenylbutazone to a patient on warfarin therapy, it can result in the hemorrhagic reaction and it can cause the displacement affinity for a particular drug. So the clinical significant interaction like the displaced drug, for a displaced drug molecule, it is more than 95% of plasma protein bound. It having the small volume of distribution. So more protein binding, it causes the less tissue concentration is available in the body. It having the smaller volume of distribution. And it show high onset of action. And it having the narrow therapeutic index as compared to the drug interaction. So when we talk about the displacer drug, so displacer drug it has the higher degree of affinity and it compete for the same binding site, and because the drug protein concentration is highly high, it show rapid and larger increase in the plasma drug concentration. So the displacer drug it causes the more significant interaction and more competitive binding towards the displaced drug. I have already discussed you the if the drug has a larger volume of distribution, it will be remain in the blood for a longer period of time and devastated adverse effects. But if the drug has a smaller volume of distribution, it will largely be distributed to a central compartment and it's rapidly secreted in the urine. So it shows you the insignificant side effects. So when we talk about the competition between the drug and the normal body constituents, so the free fatty acids available in the body, they are known to interact with a number of drugs that bind primarily to the human cerebral albumin. That leading to the increase in the free fatty acid levels that result in the diseased or altered physiological or pathological condition. Example like fasting diabetes, or that causes the after administration of caffeine, the free fatty acid level the increases. So the increased free fatty acid level it causes the improved metabolism after fasting or after the fed state when we taken the meal, because caffeine it increases the enzyme induction of various hepatic enzymes like cytochrome P450 that causes the greater metabolism when we taken the food, or it causes the decreased metabolism in the fasting state. Basically it is of concern when we talk about the diabetes patient. Fatty acid it binds with the albumin and thus it decreases the binding of the various acidic or non-ionized drugs. In the neonates, bilirubin it binds to the hemocerebral albumin and it gets impaired with various type of the acidic drugs. Thus, Free bilirubin, it reaches to the blood-brain barrier, result in the that is called kernicterus. Kernicterus basically it's a disease condition that is causes the degeneration of the brain and the mental retardation. Next type is the allosteric changes in the protein molecule. So that process that it involves the protein structure by the drug or its metabolite, thereby modifying its binding capacity. For example, aspirin. So aspirin is acetylates the lysine fraction of the albumin, that thereby modifying its capacity to bind the nascent molecule like the phenylbutazone. So basically, the active site of the phenylbutazone, it having that acetylation. So aspirin, it competitively binds to the lysine fraction of the albumin and it modifying its capacity. So the phenylbutazone concentration in the blood is highly high, so it can be rapidly distributed to the tissue compartment. So basically, there is less chances of the unsprayed form of the drug changes in the urine, and it can be available in the body for a longer period of time. Fourth type is the patient-related factors. So basically, age is one of the important factors in the neonates. If the albumin content is low, so the more free drug is available for circulation. Elderly, it's having that low albumin also, so high chances of the drug available for the circulation for the therapeutic action. So intersubject variability, so due to the genetic and the environmental factors, but what that protein binding, it can play a major role in the genetic variation. When we talk about the genetic polymorphism is there, then if the decreased or increased concentration of the enzymes in different patient conditions, so it can alter the protein binding. See disease state, so it will affect the pharmacokinetics and the pharmacodynamics of the drug. So in various disease like the renal failure, hepatic failure, or the inflammatory states like we have the trauma or surgery, albumin plasma is albumin synthesis or the albumin content is very low, so it decreases the binding of the acidic drugs, neutral, and the basic molecule that are unaffected by the systemic circulation. And when we talk about the hepatic failure, the decreased albumin synthesis is brought by the decreased binding of the acidic drugs and the binding of the basic drugs. It also depending upon the what level of AAG in a body acquired. So decreased AAG level, it also causes the decreased binding of the acidic drug to a particular protein. What we call about the inflammatory states like the trauma, surgery, so it improves the alpha acetylglycoprotein level, so it increases the binding of the basic drug, neutral, and acidic molecule that are unaffected or unchanged excreted in the urine. What is the significance of protein or tissue binding of a drug? This is the one of the very important parameter. So when we talk about the absorption, as we know that the conventional dosage form, it follows the first order kinetics. So when there is a more protein binding, it disturbs the absorption equilibrium. So if the absorption equilibrium is disturbed, then there is a disturbance of a uniform distribution of concentration in the peripheral or in the central compartment. So there is a chances of the unvariable or erratic bioavailability in the body. B is the distribution. So a protein bound drug in a particular does not cross the blood-brain barrier, the central barrier or the glomerulus. So thus protein binding, it decreases the distribution of the drug. When we talk, but in the neonates or in the elderly, because of the blood-brain barrier is not been fully developed. So it will causes the binding affinity to be increased, and it got increased the distribution of the drug in very central to peripheral compartments. So depending upon the age, so age is the one of the most important factor when we talk about the crossing the drug to various physiological barriers. But in general, a protein-bound drug is not cross the blood-brain barrier, the placental barrier, or the glomerulus. C. Metabolism. Protein binding, it decreases the metabolism of the drug and it enhances the biological half-life. So only the unbound fraction it gets metabolized. For example, phenylbutazone or sulfonamide. Phenylbutazone or sulfonamide, they are competitive inhibitors. So one drug, one binding can displace the other binding. So the displaced drug is available for the circulation for longer time due to accumulation of toxicity. So it improves the biological half-life and decreases the metabolism of the drug. D is the elimination. So only the bound drug is capable of being eliminated. because the bound fraction is pharmacologically inactive, and the unbound drug, which is basically pharmacologically active, is available for circulation and is capable of being eliminated. Protein binding, it prevents the entry of a drug to a particular metabolizing organ like the liver and to the glomerular filtration. So, for example, tetracycline. Tetracycline is eliminated mainly by the glomerular filtration. E is the systemic solubility of the drug. So, lipoprotein, it acts as a vehicle for the lipophilic drug like the steroids, heparin, or oil-soluble vitamins. So, it can improve the systemic solubility of a drug. F is the drug action. So protein binding, it inactivates the drug because sufficient concentration of the drug cannot be built up in the receptor site for the action. So basically, I have already discussed with that the unbound drug is available for the action. So protein binding, it majorly extend the binding concentration of a drug. It can pick up the concentration of a drug for binding, and it inactivate because the concentration is not sufficient, because the bound concentration is very high and the unbound concentration is very low for the therapeutic action. So sufficient concentration of the drug cannot be built up to the receptor side for the actions. For example, naphthoquinone, sustained release. So the complexity of a protein binding, it also acts as a reservoir, because the bound drug is slowly being detached from the body and it available for circulation. So this is basically a factor of a choice when we deliver a drug in a sustained release fashion, or when we convert a drug to a sustained release product. Basically, the complexity of a drug in the blood, that it play a major role to sustain the release for a longer period of time. For example, suramine. So basically, the market formulation as a sustained release is available for its anti-trypanosomal action. So basically, because the binding is very high, about 99% of the protein-bound molecule is available, and only the 1% is unbound available for a therapeutic action. So the 99% binding affinity, it creates a sustained release drug for a longer period of time. H is the diagnosis. So basically, the chlorine atom of chloroquine, it replaced with the radio-labeled I131 that can be used to visualize the melanomas of the eyes and the disorder of the thyroid gland. So basically, drug targeting, the binding of the drug with the lipoproteins, it can be used for the site-specific delivery. For example, cancer cell that having the greater affinity for the low-density lipoprotein receptors. So basically, these are the references that can be taken with the data. Okay, now we have shifted towards the kinetics of the protein binding. So if the P represents the protein and the drug, it represents D represents the drug, then we can apply the laws of mass action to the reversible protein binding. So protein, it binds with the drug, and basically it's a reversible binding and it forms the protein drug binding mixture at equilibrium. So by applying the law of mass transfer, the rate constant is get generated and is equivalent to the bracket open protein drug binding divided by protein and the particular drug. So we can substitute the value of protein drug mixture, that is PD, is equivalent to rate constant Ka multiplied by P into D, where P is the concentration of the protein, D is the concentration of the free, KD is the concentration of the free drug complex, and Ka is the association rate constant. If the PR is the total concentration of the protein present, whether it is bound or unbound, then PT is equal to PD plus P, where R is the number of the moles of a drug that bounds to a total mole of the protein. So we can rearrange the equation, that is R is equal to PD divided by PT. So basically we can substitute the value of PT, that is basically PD in the numerator, and we can substitute the value of that PT, that is a value it got we can get is PD plus P. So R is equal to, we can substitute the value of then rate constant, and Ka bracket open PB bracket in the denominator, Ka PD plus P. So we can commonize, or we can common the value of Ka, that is Kad plus 1. So the final equation we can get is Ka bracket open D, Kad plus 1. So the above equation that it holds when there is only one binding site on a protein and the protein drug complex is available in a one by one complex. If this the above equation is hold for a one binding site, if we get a number of binding site, if more than n number of binding sites is available per molecule of a protein, then R is equal to n KAD divided by KAD plus 1. So the value of the association constant Ka and the number of binding site n that can be obtained by the plotting the above equation in four different ways. So we can use the equation and we can plot the different concepts, and we can calculate the Ka and the number of binding site n by using direct plot, scattered plot, closed plot, or Hitchcock plot. So firstly, we have discussed about the direct plot. So a direct plot that is the number of the mole of the protein versus the drug. It can be used to find out the number of binding site available for a protein in a plateau phase. So Ka is obtained by finding the drug concentration that is required to saturate the half of the binding sites available. That is basically n by 2. So we can plot a value between the number of the protein molecule, that is basically R, and the drug that needs to bind to a particular protein, and we can plot a graph until a plateau phase is key. So based upon that curve, we can get the value of the n by 2, that is basically a number of the binding sites that will be available to saturate the drug, or what we call as the rate constant from the slope of a graph. Scattered plot. Scattered plot. So basically, in the previous equation that we can get that they basically R is equal to NKAD divided by KAD plus 1. So basically, we can rearrange the equation into a linear form. We can get this R plus R KA bracket open D bracket close is equal to NKAD. We can commonize the value of R is equal to NKAD minus R KAD. So basically, R, and we can substitute the value of D in the denominator, and R divided by D is equal to NKA minus RKA. So we can plot a graph between the R divided by drug versus the R. It yields a straight line with the XY intercepts, and we can get a value of N, that is basically number of binding site, and we can get a value of Ka from the slope. So basically, as I suggested you, we can plot a graph between R by D and R, and we can get a linear curve with a slope of minus Ka, and we can get a value of Nka in a scatter plot. Third is the double reciprocal plots or closed plot, that is also called a line Weber plot. So reciprocal of that equation, that is basically R is equal to NKAD divided by NKAD plus 1. So weekly reciprocal of that equation, it gives 1 by R is equal to 1 NKAD plus 1 by N. This is the equation we can get in the reciprocal form. So a plot of 1 by R versus 1 by D, it yields a double reciprocal plot. It is a straight line basically with the slope m, but it's basically 1 by nka, and y-intercept is 1 by n. Basically, a final plot is available. That is basically a plot between the 1 by r and 1 by d, and we can get a slope value that is basically 1 by nka. Third, fourth plot is the Hitchcock plot. So when we talk about the enzyme kinetics, when we talk about the linear curve rhythm, so this plot is widely being used. So in the above equation, that is basically R is equivalent to NKAD divided by 1 plus KAD. So we can rearrange that equation into a linear form again. It is basically NKA bracket open D divided by R is equivalent to 1 plus KA. So we can divide the both sides by NKA. We can get the D, what we call as the divided by R is equal to 1 divided by NKA plus D divided by N. Or D by R versus D, it yields a straight line with the slope 1 by N and intercept 1 by NKA. So this is the basically outline of a Hitchcock plot. Basically, a dividend of D by R versus D, and we can get a slope value of 1 by N. So this data I have already taken from the biopharmaceutics and pharmacokinetics biomarker. So thank you very much for your attention. So any suggestion, any detail, you can contact me at a listed address. Thank you.